substrate A. I think I'm the perfect match for you.
Okay, kailangan mo kasunod. Go.
Wait, pasod lasad ko!
Oops!
Ayos na, Mandy.
Uy, saan mo to? Hindi mo to? Ito. Get out!
Iba ko nakulang ko sa kong shape, pero mo-try daw ko. Di ba mo-adjust na ka?
Okay. Tira ka masud. Oh, sama na.
Pinakapasunod ako. Uy, masud ko. Dili. No.
Sama na. Uy, si Ami dira. Uy!
Good day, everyone. Today we're going to explore a fascinating and essential topic in biochemistry, the relationship between protein structure and function, specifically focusing on enzymes and the models that explain how they work, the lock and key model and the induced fit model. So these models help us understand why enzymes are incredibly specific and how even tiny changes in structure can drastically affect how they function. Not only is this concept important in biology, but it also has real-world medical applications from drug design to diagnostic tests. So, let's get started! First, what is an enzyme? Enzymes are proteins that act as biological catalysts. That means they speed up chemical reactions in the body without being used up themselves. So almost every chemical reaction in our cells depends on enzymes to occur fast enough to sustain life. Now, what do enzymes act on? So they act on molecules called substrates. A substrate is simply the reactant that an enzyme binds to and transforms into a product. Think of it like this. If the enzyme is a machine on an assembly line, the substrate is the raw material it works on. So the machine, the enzyme, changes the raw material to substrate into a final product. So it's important to understand that enzymes are highly specific. One enzyme usually works on one particular substrate or a very small group of closely related substrates. This brings us to the idea of enzyme specificity, which is explained by two important models. The first model is called the lock and key model, proposed by Emil Fischer in 1894. According to this model, the enzyme has an active site, a specific region where the substrate binds. The shape of this active site is exactly complementary to the shape of the substrate. Just like a key only fits into a particular lock, only the correct substrate will fit into the enzyme's active site. Let's look at this diagram. Notice how the substrate fits perfectly into the enzyme. That's the idea of the lock and key model. Now answer this. What would happen if the substrate shape didn't match the active site? Exactly! It wouldn't bind and the reaction wouldn't happen. This model helps explain the specificity of enzymes, but it's a bit too rigid. In reality, enzymes are flexible. That brings us to the next model. Next is the induced fit model proposed by Daniel Koshland in the 1950s. It's a more accurate representation of how enzymes really work. So according to this model, the enzyme's active site isn't the perfect fit to the substrate right away. Instead, when the substrate begins to bind, the enzyme changes its shape slightly to hug the substrate more tightly. You could think of it like a hand putting on a glove. The glove may not perfectly match your hand shape at first, but as you wear it, the glove molds to your fingers. The enzymes behave the same way. It adjusts to fit the substrate. So this flexibility increases the enzyme's efficiency and helps explain how some enzymes can act on similar substrates with small differences in structure. Answer this. Based on what we've seen, which model do you think is more realistic in biological systems and why? Answer it in your notebook. Let's take a moment and imagine this. What would happen if enzymes weren't specific? Picture the digestive enzyme pepsin, which breaks down the proteins in your stomach. What if pepsin suddenly started breaking down your stomach lining instead of just the food proteins? That would be disastrous. Enzyme specificity prevents this from happening. It ensures that enzymes only catalyze the right reactions with the right molecules. This is especially critical in metabolic pathways, where one wrong reaction could lead to the accumulation of toxic substances or the failure to produce vital compounds. So enzyme specificity isn't just about efficiency, it's about safety and survival. Now let's talk about enzyme inhibition. So sometimes enzyme activity needs to be slowed down or stopped. This can happen naturally with the help of drugs. There are two major types, competitive and non-competitive. So competitive inhibition is where an inhibitor molecule competes with a substrate for the enzyme's active site. It's like two similar keys trying to get into the same lock. And non-competitive inhibition is where the inhibitor binds to another part of the enzyme, changing its shape so the substrate no longer fits well. We also have reversible and irreversible inhibition. In reversible inhibition, the inhibitor can detach and the enzyme goes back to normal. In irreversible inhibition, the enzyme is permanently deactivated. This concept is the basis of many medications. For example, some antibiotics inhibit bacterial enzymes permanently, which is how they kill bacteria. Now, let's connect all this to real life. In medicine, understanding how enzymes work helps scientists design better drugs. If we know the exact shape and structure of an enzyme involved in a disease, we can develop molecules to block or alter its function. For example, HIV protease inhibitors block a viral enzyme that's essential for HIV replication. Aspirin works by inhibiting an enzyme involved in pain and inflammation. COVID-19 treatments also target viral enzymes. Even in diagnostic tests, enzymes are used to detect certain molecules in blood or urine. Answer this. Can you think of any other cases where understanding enzymes might help in health or disease? Let's wrap this up with some reflection. Think about these questions. What happens when a protein like an enzyme is folded incorrectly? Why do enzymes only bind to specific substrates? And what is the key difference between the lock and key and induced fit models? And can you explain the difference between reversible and irreversible enzyme inhibition in your own words? I'd like you to write short answers to these in your notebook. To summarize, enzymes are specific biological catalysts that bind to substrates based on their structure. The lock and key model shows the perfect fit, while the induced fit model reflects a more flexible and accurate mechanism. Enzyme specificity ensures biological reactions happen correctly and safely. Enzyme inhibition is not only a natural regulatory mechanism but also a major tool in medicine and drug development. That's all for today. Thank you.